Good evening, everyone. Hi, welcome. Hi, look, I'm Pankaj Shah, the director of the QBI. And so, look, welcome everyone to, I think it's probably our 8th Merson lecture. Started in 2008, and we've had these, and a couple of them we didn't have. Of course, we stopped for COVID, but finally we're launching them off again, which is great. And it's fantastic to have Eric Nestler here, who've been trying to come here for a few years now, I might add. And so look, I'd like to begin by acknowledging the traditional owners, the Jagera and the Turrbal people and their elders who have looked after this land for many thousands of years, and I'm sure their new generations of elders who are coming will continue to do so, assuming we actually pay attention to what they tell us, and, but they've done a fantastic job. And look, it's great to have these meetings again, finally starting this year. And this lecture was started by the generosity of David Merson, who you will hear from a bit later. And David started the Merson lecture series. And this is a lecture series which has been, as I said, going for about 8 years. And it really is a public lecture, unlike a very specific, we have a few other lectures which are more science-based, but this is really to give the message and tell the people in the community what neuroscience is about, and the kinds of things it does, and why it should be paid attention to by everyone. And of course, this is the QBI, and the brain and what it does is pretty amazing. And why would you not want to work on the brain? It is really the most exciting place there is. And Eric and I are both, and we're very similar in our backgrounds in many ways, and really think about why it is that the brain is so amazing and the disorders that come from it. And we all know about dementia, but there's a range of other things that the brain does, including the reason you're here, no doubt. So, look, Eric started in medicine, I guess not really. He did a BSc and he trained at Yale University. He did a PhD and then medicine. He did an MD PhD program, which is a very strong program in the United States. We do have this here in Australia as well, but not as many people go through the program. But an MD PhD program, you sign in for this program as an undergraduate, and at the end of it, you come out with a PhD as well as an MD, which is like our MBBS. After which, Eric decided to do a PhD, and then with Paul Greengard, and some of you might know Paul Greengard got a Nobel Prize and was involved in the dopamine system and really interested in phosphorylation of proteins in the brain. So Eric then trained in psychiatry. Maybe it's the other way around. He was trained in psychiatry first, and at the end of the psychiatry training, did a PhD in neuroscience, and pretty much has stayed in neuroscience since then. So he's currently the Nash Family Professor of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York. And if any of you visited there, Mount Sinai is an amazing place with a lot of neuroscience going on there. Very exciting place. He also serves as Dean for Academic Affairs, Chief Scientific Officer, and Director of the Friedman Brain Institute. Just a few things to keep himself busy. So after his PhD and MD degrees, he finished psychiatry training, and then he served on the Yale faculty from 1987 to 2000, where he was Elizabeth Means and House Jameson Professor of Psychiatry and Pharmacology and Neurobiology, and Director of the Division of Molecular Psychiatry. So from there, he moved to Texas, where he was at Southwestern University, and he was the Lou and Ellen McGinley Distinguished Professor and Chair in the Department of Psychiatry there. He then moved to New York, which is where he was born, and went back to, I guess, his place where he spent all his early years, and has been there for the last 8 years or so. Eric is someone that, I've read many of his papers over the years, and I'm sure many of you have had. He's a distinguished career in publication with over 500 articles, and really covering many aspects of things that we think about at QBI as well, and has covered many areas, but he has really been interested in second messenger signaling and phosphorylation of proteins, and more recently, really thinking about the kinds of areas that come from his psychiatry training. So, Eric, it's a pleasure to have you here. We're looking forward to your talk.
Thank you. Fantastic. Pankaj, thank you so much. It is such a treat to be here in Australia. It's my wife's and my first time to be down under, and we're really excited to be here. Um, I am the director of the Friedman Brain Institute, and I think even in Australia, you could imagine what it means to be the director of the FBI. And that's something that I've enjoyed a great deal over the last number of years. What I'd like to do today is to tell you about depression from my perspective, from a point of view that will be accessible to the lay public, and I'll be focusing on mechanisms that we will term transcriptional and epigenetic, and for those of you who are not scientists, part of my job today is to help you understand what I mean by those terms. So let me just begin by putting the context of depression, providing some context for depression. Here we are in 2023, and we still diagnose depression and all other psychiatric syndromes solely on the basis of behavioral abnormalities. There is no blood test, brain scan, genetic test that assists in the official diagnosis of depression or any other psychiatric syndrome. That's astounding when we compare this to every other branch of medicine, especially given that depression is now the #1 cause of disease burden worldwide. It's more common in women than in men, but even in men, it is the #1 cause of disease burden. Estimates of how common depression are vary quite a bit. 3 to 5% of severe depression. It's been estimated that perhaps more than a quarter of all humans on Earth will suffer from more mild cases of depression at some point in their lives. And this incidence of depression has increased dramatically since the COVID-19 pandemic, we think due to the social isolation and economic dislocation that people have suffered. We still know relatively little about what causes depression. I view depression as one of several human stress disorders, along with post-traumatic stress disorder and anxiety disorders. I'm not sure what differentiates those three conditions from one another. As I said, there is no biological measure that distinguishes those people. It's all based on a different range of behavioral abnormalities. But in parts of the world that suffer tremendous trauma, roughly 1/3 of the people who seek psychiatric attention are diagnosed with depression, another third PTSD, and another third of anxiety. So I would view them as highly similar conditions, and we know they're also very comorbid, meaning they occur in many people at the same time. I'll be saying a little bit more later, but both these conditions are caused by a combination of genetics and we think by exposure to lifetime stress. Most importantly, we view these conditions as syndromes, meaning that it's not like saying pneumococcal pneumonia, that's a disease. Cough, fever, chills is a syndrome. Depression, anxiety, PTSD are syndromes. They're heterogeneous collections of many disease states, and it's been a challenge to identify the specific abnormalities involved. Now, despite that, we do have some pretty good antidepressants. Right? We're all familiar with serotonin selective reuptake inhibitors and related types of antidepressants that were introduced now close to 70 years ago. They were discovered by serendipity, by luck, not based on any knowledge of biology. And most importantly, today's best antidepressants only fully treat roughly half of all people who are depressed. So let me emphasize that. This is the #1 cause of disease burden, and we can only treat half of affected people. So the unmet need in treatment of depression is enormous. It's hard to overstate. If we stopped research now, we would not be able to help half of the people with the world's greatest disease burden. So we need more research to better understand the biology of depression. Okay, so how are we going to do that? We think that stress is a major factor involved. Stress is the strongest known risk factor in the cause of all of these conditions that I talked about. Sometimes people refer to this as adverse life events or trauma. But basically what we're talking about is different forms of stress. Yet we know that most humans can remain resilient despite horrendous levels of stress. This has particularly been shown in cases of combat, for example. Also in civilian cases, people who are victims of physical or sexual assault, a majority of people can do pretty well despite being exposed to that type of stress. So we can't only look at what stress does to the human brain, but we need to understand what it is that makes some individuals more susceptible to stress and others more resilient to stress. And what's particularly important is I don't want to classify people as susceptible or resilient, because in fact one individual can be very susceptible to one type of stress but quite resilient to another. What we've learned over the years is that this question of stress susceptibility and resilience is controlled by the combination of an individual's genes and his or her life experiences. We call this gene by environment interaction. So let me just drill into that a little bit more. About 35% of the risk for depression is heritable. Slightly less for PTSD and anxiety. We talk about the genetics of depression, of psychiatric syndromes being complicated, but it's really complicated for these human stress disorders because that 35% of risk is comprised by many hundreds, perhaps even thousands of genetic variations. A human only has 20,000 genes. So to say that 1,000 genes contribute to the risk for depression is being pretty vague, because each of these 1,000 genes contributes a minuscule amount to that total 35% risk, which is why we can't do a genetic test today to tell us what our risk is for depression. The remaining 65% of risk is due to life experiences, beginning in utero and extending throughout life, and again with stress being a major contributor. I'm going to stop using the touch screen because I'm having a problem. I'll use the laser pointer. The notion of gene by environment interaction is crucial because it means that genetics and life experiences are not deterministic. That when a person has a low genetic risk for depression or these other syndromes, they still can get sick if they're unlucky enough to be exposed to a lot of unfortunate experiences in life. Conversely, a person who might have a very high genetic risk may remain healthy throughout life if they're lucky enough to avoid serious stress. So one way to approach human stress disorders is to better understand the basic biology underlying this inherent susceptibility versus resilience to stress, and to use that basic neurobiology to develop improved treatments. And when I refer to treatments, I'm talking about better medications, but also non-medication treatments. For example, there's been a great deal of attention given recently to mindfulness, meditation, hypnosis, things like that, as well as psychotherapy. Most of the field has focused over the years in preventing or reversing the bad effects of stress. A relatively new notion is, what about recapitulating mechanisms of natural resilience? If we could understand what makes some individuals more resilient naturally, maybe there are ways to institute in that individual's brain those natural resilience mechanisms and boost their resistance to stress-related disorders. I'll come back to that in a bit. I mentioned the heterogeneity of depression, and I wanted to describe a bit more about that. This comes from a figure by Connor Liston, a friend and colleague in New York. What it is showing is the heterogeneity of depression. In a population of people who are depressed, depression comes in many different forms. In fact, as a psychiatrist, I could diagnose the same syndrome of depression in two individuals who have no overlapping symptoms. How could that be the same illness if people do not have the same symptoms? Okay, so if there were a way to parse the broad depression syndrome, perhaps we could do a better job at developing treatments. After all, a person with the green type of depression might respond to one type of treatment, and a person with the blue type of depression might respond to a different type of treatment. So there's been a tremendous effort in developing biologically based diagnostic tests for depression, a work in progress. We haven't gotten there yet, but you are making some progress. These efforts focus on genetic variation, what's called so-called polygenic variation or polygenic risk, analyzing thousands of genes at once based on our genome sequence, which will become available for all of us in time, to determine a risk for depression, but not only depression, a certain type of depression. To combine that with knowledge or ascertainment of brain circuits through use of advanced brain imaging approaches, more complicated behavioral analyses of a person's clinical symptoms, and so on. Work in progress, not quite there yet, but I think that this will be the future in diagnosis and treating psychiatric syndromes, just the way we do in the rest of medicine. This slide depicts a strategy by which we can do that in an integrative way by combining studies of animals and humans across multiple levels of analysis. And let me describe what this slide is depicting. Let me start with genetics. I'll say a bit more about this in a few minutes, but genetics is encoded in our DNA, which you'll recognize as the double helix. I mentioned that a human has about 20,000 genes. Those 20,000 genes come together to determine the traits of individual cells in the brain. Individual cells in the brain communicate to form nerve circuits. Nerve circuits in the brain control behaviors, and we're using advanced artificial intelligence to understand how functioning of brain circuits control a behavior. The importance of doing this across species is that humans are what we care about. We want to understand and solve human disease, but the ability to do experiments on humans is legitimately very limited. By and large, the sole way that we can study mechanisms is in animals. So we think that the study of laboratory animals is an essential component in this effort, the way it's been for the rest of medicine. Any medication, almost any medication, 99% of medications that any of us are on were developed through advances made in animal research. I'll be happy to answer any questions about that during the course of after the lecture. So you'll notice the arrows connecting these various levels of analysis. The genes control functioning of cells, cells control functioning of circuits and behavior, but every time electrical activity is firing within circuits in the brain, it's changing the degree to which genes are turned on and off. And that process is named epigenetics. So the epigenetics can be viewed as the mechanism governing the interplay between genes and environment. And that's why it's so important. Let me say a little bit more about epigenetic regulation. An individual starts life as the fusion of a sperm cell with an egg cell. Epigenetics begins at that moment of life. As that single fertilized cell develops into a fully differentiated organism, including a fully differentiated brain, trillions of cells are developed in the body, and in the brain, intricate connections are made among those cells. And a lot of what happens during development is stochastic. It's random. That is referred to as epigenetic variation. That is why identical twins have different fingerprints. It's just a random process. Identical twins have different patterns of gyri, the folds, on the surface of the brain, because that too is a reflection of epigenetics. Once an individual is fully developed, epigenetic mechanisms respond mainly to diet and lifestyle, but a whole range of other environmental exposures, in particular for today's lecture, stress throughout life, from prenatal stages to childhood to adulthood and to advanced age. Our brain is continually being modified through gene-environment interactions, through epigenetic mechanisms. Now let's move on to the brain and understand where in the brain depression might be arising. Unlike Parkinson's disease, which is largely, to a large extent, caused by the death of one type of cell in one brain area, depression's pathology seems to be far more distributed across the brain. We refer to these regions as limbic regions, the emotional circuits in the brain, which involve broad regions of the prefrontal cortex, which has been particularly evolved in humans and non-human primates. A brain area called the nucleus accumbens, which is the major reward center in brain. Two brain areas called the amygdala and hippocampus that are located in the temporal lobe on the side of the brain, particularly involved in memory and emotional behavior. And the hypothalamus, which controls the body's physiological functions. All of these regions are innervated by neurons located in the brainstem that make what are called monoamine neurotransmitters. Examples include serotonin and norepinephrine, and almost all of today's antidepressants work on those two neurotransmitters. That innervation of these four brain regions by these monoamine making neurons gives those antidepressants their mechanism of action. Now, a single human brain is really complicated. We know that a single human brain has on the order of 100 billion nerve cells and 100 billion non-nerve cells called glia cells. The glial cells mainly are thought to serve as supporting cells to modify the functioning of the nerve cells. I mentioned that nerve cells come together to form circuits. Each nerve cell, in fact, can connect with many thousands of other nerve cells. So it's been estimated that a single human brain can have 100 trillion or so connections. And that's an astounding number. Albert Einstein estimated a number of stars in the universe. That 100 trillion is a bigger number. Okay, that's a single human brain. A single human brain can do some things better than the world's most advanced computer. And we're still at the very early stages of cracking how that works. Deep brain stimulation, which I know is a major research interest here at QBI, has provided a rare example where one can obtain mechanistic data from a human brain. Deep brain stimulation is now a very well accepted treatment for Parkinson's disease and other movement disorders, but has been used experimentally to treat depression and other psychiatric syndromes. Work by Helen Mayberg and colleagues at Mount Sinai and many other places. Deep brain stimulation is sort of like a cardiac pacemaker, but instead of putting an electrode in your heart, you put an electrode in a precise part of the brain. The electrode is turned on, and delivery of electricity to certain parts of the brain can make a person who has failed getting better with chemical antidepressants get better. And it has provided experimental proof that these brain areas that I glibly said are related to depression, proving that they really are, because deep brain stimulation of a region of the prefrontal cortex or a region close to the nucleus accumbens can alleviate depression. Now, these are individuals who are so severely depressed that a neurosurgical approach becomes feasible and justified. Nevertheless, it provides critical information. But to gain more mechanistic information, we need to look at laboratory animals, just like in every other branch of medicine. But here in psychiatry, we have a particular challenge in the use of laboratory animals. Because the very symptoms that we use to diagnose a psychiatric syndrome are mostly inaccessible in any animal, right? And so this is no doubt one reason why progress in psychiatry has lagged behind that of other medical disciplines, just because it's so difficult. We can't measure blood glucose in a mouse as a measure of diabetes. We can't measure cardiac output as a measure of heart disease. We have to measure these behaviors, right? And that's really difficult to do in a rodent. So let me now tell you about one model that we've used that involves chronic stress that we think is relevant to human stress disorders. This was developed in the lab by Olivier Berton, who is at NIDA, National Institute on Drug Abuse at NIH, and Nadia Tsankova, who's at Mount Sinai. And what they did was to take a C57 genetically inbred mouse, just a standard laboratory mouse, and they put it in the cage of a bigger, meaner mouse, a retired breeder CD1 mouse. Fighting ensues instantaneously. We limit the fighting to a few minutes, so to limit physical injury. And then we house the test mouse across from the aggressor mouse with a screen so that sensory contact continues for the rest of the day. And we repeat this in a standard protocol every day for 10 days. This test mouse is exposed to a new aggressor mouse. At the end of that 10 days, it's a horrendous level of stress, mostly psychological stress for the mouse. The mouse develops a behavioral syndrome, right? It's a mouse stress disorder, and these are the symptoms. We say that the mice have anhedonia. That means decreased interest in pleasurable activities. Actually, that's something I think we can measure pretty well in an animal. These mice show much less interest in consuming sugar water, eating high-fat food, having sex, running on an exercise wheel, and so on. We say the animals have anxiety, but I don't know how I'd ever know whether a rodent is anxious or not. The rodents just explore their cages to a lesser extent. They become more cautious in their environments. They have hyperactivity of the hypothalamic pituitary adrenal axis. That means they secrete more glucocorticoids. In humans, that's cortisol. They show disrupted circadian rhythms. They're more vulnerable to become addicted, to addict themselves to cocaine, opioids, and other drugs of abuse. They develop a metabolic syndrome where even though they seem to like eating less, they eat more, they become obese. They induce obesity in themselves. And that's interesting because about half of people with depression are overweight, and there's a higher incidence of depression in people who are overweight as well. And they exhibit profound social avoidance. I'll come back to that in a minute. Now, only about half or so of the mice exposed to this paradigm develop that full range of behavioral symptoms. We call them susceptible. A smaller subset, just under half, avoid all of these other behavioral abnormalities. They still show the reduction in exploratory behavior, the anxiety-like symptoms, but avoid all the rest. We call these mice relatively resilient. So this makes social defeat a particularly interesting stress procedure because it's one of the few that can differentiate a mouse's response to either succumb to the bad effects of stress or to remain resilient. Some of the symptoms, particularly these ones related that are avoided in the resilient animals, are essentially lifelong. We've looked at these mice up to 6 months after the 10 days of social defeat. They're unchanged. They can even increase over time. And we can show that these symptoms can be treated when we give the mice repeated administration of a standard antidepressant like an SSRI, and we know that in humans, SSRIs only work after repeated administration. Single doses are not antidepressant. Anxiolytic benzodiazepines are without effect, and single doses of ketamine are also effective. And I think that ketamine is not yet officially approved for use for depression in Australia. It is in the United States, and we think it is a reasonable treatment for someone who has not responded well to more standard antidepressants. Okay, so with that as an introduction, I'm now going to dive into more biology, and I'm going to focus on two principles that we've learned from using this mouse model. First, I'm going to tell you more about the dramatic differences in the biology of stress-related illnesses in males versus females. I mentioned that depression is twofold more common in girls and women than in boys and men. There are mild differences in the clinical syndromes, some differences in symptoms, some differences in treatment responses, but the clinical syndromes are more similar than different. Yet little is known about the underlying molecular mechanisms of the greater vulnerability, on average, of females than males. And then after that, I'll give you an example of how we've provided novel mechanisms and some leads for new treatments for depression by diving into the molecular basis of stress resilience and susceptibility, work that can only come from the study of animal models. And I'll introduce a gene, doesn't matter what the gene stands for, ZFP189, which we've identified as a novel pro-resilience gene. Okay, so let me go back to this cartoon and describe epigenetics in a little more detail. I mentioned that a human and a mouse have about 20,000 genes. Those genes are encoded in linear spans of the DNA double helix. Each of these steps in the ladder is called a nucleotide or a base. A mammalian genome has 3 billion nucleotides. If stretched out linearly, those nucleotides would be about 2 meters in length. In every cell in the body, somehow 2 meters of DNA fit into a microscopic cell nucleus. That packaging of the material inside of a nucleus, the material is called chromatin, requires extensive condensation of the DNA. We've understood now how that occurs. The DNA double helix is wrapped around octamers of histone proteins. Histone is just a name given to a protein. Two wraps of DNA around an octamer of histones is called a nucleosome, the unit of chromatin. Those are then further organized in very rote ways in the most condensed fashion to what is recognized as a chromosome within the nucleus. This organization is essential because a nucleus is not big enough to have all 2 meters of DNA stretched out. Most DNA in a nucleus has to be condensed in order to fit, and only small pieces of DNA are open when the DNA is needed to encode a protein. That becomes really important because proteins can only be encoded by DNA, transcribed from DNA, when they're in a relatively open state, whereas more condensed chromatin is inactive. So by studying the structure of chromatin, we can help identify the genes that are affected in mice or in humans by stress exposure. We can understand the mechanisms at the level of transcription by which those changes in gene expression occur. Transcription simply means transcribing DNA into the encoded RNA and then into protein. And we believe that epigenetics provides a unique mechanism of lifelong adaptation. So the field of epigenetics started in development, as I mentioned. During development, when a stem cell is differentiated into a liver or brain cell, that occurs through the selective condensation or opening of chromatin. Permanently, for life, in that cell, always a liver cell, in the other cell, always a brain cell. Same thing with cancer. When cancer occurs, when epigenetic changes occur in a cell nucleus, where the cell loses its control and just continues to divide until it kills the organism. Our hypothesis has been that epigenetics may do the same thing for behavioral experience. That one of the ways in which that we adapt or maladapt to environmental changes involves epigenetic modifications that are equally long-lived. Epigenetic modifications simply mean chemical changes that are induced in the histone proteins, in the DNA itself, many other nuclear proteins that turn genes on and off. All right, so let me now show you some data. This is an experiment, a study led by Rose Baggett. Rose actually is originally from Australia. She's now on faculty at McGill University in Canada. And what Rose did was use a method called RNA sequencing, which is a modern technique that allows us to quantify levels of RNAs, the products, of all 20,000 genes in the genome. Something that wasn't even possible 10 or so years ago. What I'm showing you here are called heat maps. Each vertical line in a heat map is a single RNA. Yellow means that there is a higher level of the RNA. Blue means there's a lower level of the RNA. I'm showing you data from 4 brain regions that I've mentioned already: nucleus accumbens, prefrontal cortex, amygdala, and hippocampus. And what I'm showing you are how the genes that are upregulated in yellow, downregulated in blue in resilient mice after 10 days of chronic social defeat stress, and how those same genes are affected in susceptible mice across these brain regions. The Venn diagrams show the same data, showing the total number of genes that are up or down regulated in the two conditions, resilience and susceptible. And when we did this experiment, we hypothesized that resilient mice are resilient because they failed to show a lot of the bad effects of stress. But in fact, that was not the conclusion across most of the brain regions. For example, in the nucleus accumbens most dramatically, many-fold more genes actually change in resilient animals than in susceptible animals, raising the novel idea that susceptibility may be the failure of plasticity, that resilience is the adaptive plasticity that enables an organism to cope positively with an adverse life event. Again, supporting the idea that I mentioned earlier, that if we understood those mechanisms of resilience, maybe we'd find novel mechanisms of antidepressant activity. Because of the legitimate questions about the reliability of animal models, how do I know what's happening in a stressed mouse is relevant to human brain, we've carried out similar data on postmortem human brain tissue. This is a study done by Benoit Labonté, who's now in Quebec City in Canada. And what he did was to do RNA sequencing, same method, on 6 brain regions of depressed humans. These are individuals who died suddenly from a car accident or suicide, whose family members generously donated the individual's brains for science. And we had the opportunity, the privilege to study their brains. We analyzed 6 brain regions that are discussed here. The BA stands for an abbreviation of different regions of prefrontal cortex. There's nucleus accumbens that I've mentioned previously and other brain areas. But the most dramatic effect that we've seen in this data set is a tremendous difference between males and females. Notice only 5 or 10% overlap in the gene expression abnormalities seen in depressed men versus normal men as compared to gene expression abnormalities seen in depressed women versus normal women. Astounding. This was not expected at all. In order to make sure that we were not being fooled in some bioinformatic complexity, we carried out a similar experiment on mice. This is a different chronic stress paradigm. This is called chronic variable stress, exposing mice to different forms of stress. And we found something very similar across brain regions, that genetically inbred mice exposed to the same stress for the same period of time that develop the same behavioral abnormalities show minimal overlap in their gene expression abnormalities, suggesting that stress-related disorders are fundamentally different between males and females. The data sets also allow us, in fact, to go back and ask the question, are our data from mice relevant? Are they validated in humans? And indeed they are. These Venn diagrams are not meant to be quantitative, just heuristic, but what we were able to do is to compare the effects of several different chronic stress paradigms in mice to data found in postmortem brain tissue of humans in 2 brain regions that I've been highlighting already. And in each case, what we found is that each chronic stress paradigm recapitulates about 25% of all the gene expression abnormalities seen in depressed humans. Interestingly, each chronic stress paradigm produces a relatively poorly overlapping set of gene expression changes. In other words, changes seen with social defeat are different than chronic variable stress and so on, suggesting that each form of stress is recapitulating largely a different subset of abnormalities. I think that's consistent with depression being such a heterogeneous syndrome. And this type of convergence is not seen when we compare published data sets on other psychiatric syndromes like schizophrenia, bipolar disorder, and autism, which we do not consider as stress syndromes. Even though they can be affected by stress, they are not caused by stress. They're much more heritable. We do not see this convergence. Okay, so then how do we take this information and obtain new insight into the biology of depression in a way that might be actionable for new therapeutics? So the strategy that my lab and many other labs have used over the years is to take the genome-wide data sets from mice. These genome-wide data sets are called transcriptomes because it's all the transcripts, all the RNAs expressed in a given brain area from resilient and susceptible mice. Overlay them, as I've shown, with similar transcriptome maps from depressed humans to ensure the relevance of the mouse findings. We used advanced bioinformatics, and I am old enough that I learned mathematics on a slide rule, but the artificial intelligence and machine learning available today is astonishing, and it's really driving this research. We use the bioinformatics to deduce networks of genes that seem to work in concert in a given brain area, in a given cell type. And within those networks, we can identify the hub genes, the most important genes that drive the biology. And then we could use the tools of molecular neuroscience to manipulate those hub genes and study causality, proving that proving the bioinformatic deductions that the hub genes really do mediate the behavioral abnormalities. This is something you can only do in laboratory animals. You cannot do this in mice. And it's essential if we want to advance this work to new treatments. Okay, so let me tell you about one gene. I mentioned before, ZFP189, never before studied in brain. It is enriched in a type of nerve cell in the prefrontal cortex called pyramidal neurons. It's the most important neuron in that brain area. And it's the strongest hub gene in a gene network that was bioinformatically ranked the strongest one related to resilience and unique to resilience. This is what a gene network looks like. The lines just show correlations in expression levels of hundreds of genes within this network. You can see ZFP189 is the strongest hub gene. When we went back to our human data sets, we found that in fact, ZFP189 is present in the prefrontal cortex at lower levels in depressed humans compared to normal humans, an effect that was equivalent in this case in men and women, an example of convergence between the two sexes. Whereas in the mice, ZFP189 is decreased in susceptible mice, consistent with that finding in humans, but induced in resilient mice. So is ZFP189 mediating resilience? Well, for that, we use viral-mediated gene transfer. We take a virus, in this case we use what's called a herpes simplex virus, doesn't really matter. We express a control protein called GFP, green fluorescent protein, it's inactive. Or we can have the virus encode ZFP189. The virus only expresses in nerve cells pick up the virus, and that way we can transfer the gene, ZFP189, into those nerve cells. We can make those nerve cells make more ZFP189, mimicking what's seen in resilient mice. This is showing results in two simple behavioral tests, although we've done it in many others. One, we can look at social interaction. In control mice, you can see not much difference between in social interaction behavior. Absent meaning there's no mouse for the test mouse to interact with. Present means there is a mouse for the test mouse to interact with. And ZFP189 really has no effect in a normal mouse. However, after social defeat stress, we see a dramatic reduction in social interaction in the defeated mouse treated with the control vector, control virus, and we can completely prevent that pro-susceptible effect by increasing the amount of ZFP189 expressed in nerve cells in that brain region, a pro-resilient effect. Same thing is found in the degree to which a mouse wants to drink sugar water, that ZFP189 is increasing sucrose preference. But most importantly for treatment is to take a mouse, stress it, subject it to stress, it develops a behavioral abnormality, and see if manipulating ZFP189 can actually correct those deficits, right? That would be required for clinical translation. And in fact, this is exactly what we found. That all of these mice were now subjected to social defeat stress. So you see the reduction in social interaction, both before this control virus. The control virus itself has no effect. The symptom is stable. Take other groups of animals, subject them to social defeat stress. You see the decrease in social interaction. But now when you treat them with the ZFP189 expressing virus, you can reverse that behavioral abnormality in terms of social interaction and also in terms of sucrose preference. Not only does it correct the behavioral abnormalities, it corrects the transcriptome abnormalities. So we can do RNA sequencing, this method to analyze all the RNAs present in a tissue, in the same animals that I just showed you behaviorally, and show that the genes within this bioinformatically deduced module are the ones that are most affected when we overexpress ZFP189 in medial prefrontal cortex neurons. We can gain some insight into how ZFP189 is doing this, how it is exerting this pro-resilience effect by recording the electrical activity from these mouse prefrontal cortical pyramidal neurons, and you could see that overexpressing ZFP189 decreases the excitability of these cells. The number of spikes, action potentials, the number of electrical impulses that are generated as you inject more and more current into a cell, you can see a much smaller response in the ZFP189 expressing cells, and this is consistent with what we've seen in other systems. First of all, many other pro-resilience genes exert the same effect in prefrontal cortex. It's consistent with our ability when we directly inhibit the activity of these PFC pyramidal neurons, that also promotes resilience. And in fact, increasing evidence supports that deep brain stimulation of the homologous region in human brain is antidepressant by suppressing ambient neural activity around the stimulating electrode. So what are the next steps? Advancing ZFP189 to the clinic is going to be really hard, and it's going to take a long time. This work was just published in 2019, and I'll show you in the next couple of slides how long it may take. I should have mentioned that this work was done by Zach Lorsch, who's a medicine resident at Duke, and Peter Hamilton, who's on faculty at Virginia Commonwealth University. So ZFP189, what does the protein do? It's a transcription factor. A transcription factor is a type of protein that binds to DNA and controls the expression of other genes. So one therapeutic approach would be to actually make a medicine that turns on ZFP189. That actually has become feasible because more and more cancer therapeutics are targeting transcription factors. So this is something that we're interested in doing and have embarked on that course of action. An alternative approach, though, is that since ZFP189 is a transcription factor, we could also make perhaps novel antidepressants by identifying which of its target genes are mediating these effects and then target those proteins for therapeutics. We're also interested in doing that. But this is a really tough challenge, and I want to highlight our experience with cancer. When I was in medical school 45 years ago, my professors told us that we would fight cancer, we would cure cancer by cutting out a person's tumor, profiling it molecularly, making an antibody or drug against a cancer-causing mutation, cancer-causing mutant protein, and it's taken 50 years to start getting there, right? We are now just beginning to see that approach taking hold in cancer drug discovery efforts. So this is the nature of making new medicines. It's not easy. Let me illustrate that difficulty in just the remaining few slides with another project of ours that has been going on in parallel that also illustrates some of the timelines involved. So back in 2007, my colleagues and I did a similar type of experiment on another brain area called the ventral tegmental area. I mentioned it earlier. It's the source of dopamine to the forebrain of mammals. And we found, just like we did in some other brain regions, that many-fold more genes are affected in resilient VTA than in susceptible VTA. And among the most different genes affected in these two conditions are a series of potassium channels that are induced in the resilient mouse VTA. Potassium channels control the electrical excitability of nerve cells. So this was an interesting finding, led us to postulate that the electrical activity of VTA neurons might actually control susceptibility versus resilience. So that led to a number of studies, starting out in our 2007 study, where we used viral gene transfer, all the way up to 2016, where we were able to show first, using viruses to overexpress the potassium channel, that did in fact exert an antidepressant effect. It turned out that this class of potassium channel is targeted by a medication called ezogabine that's actually approved for use in the United States to treat epilepsy. It's almost never used, but it is approved for use, and it has the action of activating this type of potassium channel that's induced uniquely in resilience. And lo and behold, when we gave ezogabine experimentally to a mouse, it exerted a similar type of antidepressant effect. And we gave this drug systemically as if it were a medication. So that was enough to prompt my colleagues in a study led by James Murrow to do a study of ezogabine as an antidepressant in humans. So this was published in 2019. Open label study, so you have to take it with a grain of salt. Small study, 18 people only, but treating them repeatedly, daily with ezogabine led to a reduction in their depression symptoms using behavioral rating scales, and it also correlated with a correction in a brain imaging abnormality evident in the reward circuit in the brain focused on the nucleus accumbens. This was positive enough that it led the National Institute on Mental Health to fund a double-blind, placebo-controlled study of ezogabine, which confirmed antidepressant activity. So this is a gold standard. It's a larger study now involving 40 patients and controlled, showing again a significant reduction in depression symptoms across several behavioral domains and a correction of a brain imaging abnormality, meaning that we could actually take depressed individuals, see if they have this baseline depression, brain imaging abnormality, and only those who do, treat them with ezogabine. But this was easier than ZFP 189 because ezogabine already existed. It was already approved by the US FDA. So if you started out by making a new ezogabine, it would add a decade or longer to this process. So let me just conclude with a few remarks, and I'd be happy to answer questions that you have. I hope I've convinced you that it is possible to use molecular biology to identify broad transcriptomic changes in emotional centers in the brain, both in mouse models that are related to stress and in people who die with depression. I focus today on mechanisms of natural resilience. I illustrated dramatic sex differences, and we're very interested in now in understanding what makes the transcriptome so different between males and females who are depressed. But already this argues that antidepressant drug discovery efforts need to be done on a gender-specific basis. We need pharmaceutical companies to do testing that's powered to see if treatments might work in women, not in men, or in men, not in women, something that has not yet been done. I introduced the concept of epigenetics, where certain types of epigenetic changes lead to lifelong modifications in the genetic potential of an organism. We call these lifelong changes chromatin scars, and we're very interested in better understanding their nature. And the goal is to use this information to provide a guide to develop new diagnostic tests and treatments for depression and other human stress disorders. In particular, we're very excited about ways to recapitulate mechanisms of natural resilience. That's represented by ezogabine, so we have a positive phase 2 clinical study supporting that view, as well as a wealth of data now on innumerable molecular and cellular mechanisms of resilience that have come from this type of research that are now available to be mined for clinical advances, although it is a difficult slog to do that. If it weren't, we would have cured human disease by now. So let me stop there and show you where I'm from. This is actually a picture over Manhattan that was taken by a graduate student of mine a number of years ago, and it just captured our part of Manhattan perfectly. So Mount Sinai is located on the Upper East Side of Manhattan, right by Central Park. I love this picture because this is the Metropolitan Museum of Art. Looks kind of flat in this picture. And if you can see an orange-colored building right there, that's where my wife Sue and I live. So it's kind of a fun place. So thank you so much again for having us at QBI, and I'd be happy to answer any questions.
Well, thank you Alex, for a wonderful talk. And I guess navigating through a very large landscape and talking about what the difficulties are in many ways. So look, we have quite a bit of time for questions. Fred?
Great talk, thanks very
Thank you.
much. I just wondered if there was a knockout mouse of the ZFP190.
Yeah,
Is it depressed?
not yet. That's something that we're very interested in making.
And more generally, what kind of gene do this transcription factor control?
What family of genes it controls. So we're in the process of doing that. From the RNA-seq data that we have, it controls many gene ontology terms of genes, genes related to synaptic function, cytoskeletal structure, so interesting genes. There's a very interesting interplay with the transcription factor CREB that I didn't have a chance to talk about. For the neuroscientists in the audience, they'd appreciate that. But we just made an antibody to ZFP189. One didn't exist because no one studied the gene before. And we're using that now for other methods like ChIP-seq, cut and run, to directly measure what genes ZFP189 binds to.
Could it be a marker for depression?
It could be, but again, to do that, we would have to develop a small molecule ligand that would penetrate the brain, bind to ZFP189, but it's something that we're interested in doing. Yeah. Hi.
Hi, thanks for great talk. It's surprising that this is transcription factor rather than an epigenetic regulator. And I was wondering if you think within the list of genes that you've looked at, if there are also those that regulate the epigenome, like sequence of other genes that will fit.
Absolutely. Yes.
Presented.
Among the genes that serve a hub function, transcription and epigenetic control genes predominate. Perhaps that's not surprising based on what I've said. So we're very interested, examples of epigenetic related hub genes in our various modules are histone deacetylases, for example, histone methyltransferases, and many other types of epigenetic enzymes.
What's the state of play in terms of understanding the effect of behavioural interventions on epigenetic modification in the brain? And is that a possible route into modification?
Absolutely. That's a great question because just as behavior can cause perhaps damaging epigenetic changes, perhaps there are other behavioral interventions that can be adaptive. And in fact, we've become quite interested as a field in behavioral ways of boosting resilience. And I mentioned mindfulness type approaches, but there are many others as well. It's a bit harder to study in a rodent. The notion of mindfulness is a tough one for a mouse, right? So that's what makes it a challenging area. But we would certainly expect that positive behavioral experience is working through a similar type of range of epigenetic modifications as negative stress-related experiences.
About exercise?
And exercise, of course, yes. And that you can do in a rodent. And we can show that when mice are given the option to run on a rodent wheel, which they like to do, like hamsters, they boost their resilience. They are less susceptible to chronic social defeat stress, for example, and that is associated with many of the types of changes that I've described.
Yeah, in the back there.
Thank you, Dr. Nestler, for the wonderful talk. I'd just like to know that, do you think there is any evolutionary advantages for people who are more susceptible to depression? And say if we do find a small molecular way to overexpress CPF 189, what are the expected off-target effects, and what kind of impact do you think that would have on the society? Thank you.
Yes, so two very good questions. So the first one is really interesting. Think of it, think of the mouse example before we get back to human. In our social defeat paradigm, these mice are, threatened by a physical aggressor and then live with the aggressor across the screen for a day, and that happens every day for 10 days. And the resilient mice don't seem to mind that and just keep on going through life, right? So which is adaptable? The susceptible mouse or the resilient mouse? We would say the susceptible mouse is showing non-adaptive behavior because it becomes avoidant to every other mouse. So it no longer wants to procreate. It no longer interacts with other mice that it grew up with, litter mates, brothers and sister mice, and it leads to self-sustaining social isolation. So we think it's maladaptive. But it could well be that that susceptible mouse being more responsive to a point is adaptive. It's just that the degree of stress we've applied exceeds the capacity of the system. I don't know if that makes sense. And so that's the way we would look at it in humans. That I think that what epigenetics is doing in taking a constant genome, because these mice are genetically identical, taking a constant genome and generating a lot more diversity in a species, including humans, than would be encoded in a genome, right? We are guaranteeing survival of the species to a greater extent, regardless of the environmental conditions that the species might find itself in. And that a susceptible mice might well be more adaptive under certain circumstances, whereas a resilient mouse does better under other circumstances.
Thank you for a monumental talk. Following on from that last question, it seems to me that these susceptible mice, the phenotype they go on to display would be sexually selected against over time if they're not choosing to mate. So has there been work done on establishing colonies where you just constantly stress these mice and see if over time you end up with a resilient population that can be used in studies?
Yes, there have been attempts, but it's really difficult to do. What groups have done, and you can't do this in Manhattan because of this, it happens naturally in Manhattan, but not in a laboratory. But you need a room this big to have hundreds of mice or rats populate a natural habitat. And then you can expose the rodents to different levels of stress and see how they adapt. You're absolutely right. If there was a catastrophic event, the susceptible mice, the mice that we're determining as susceptible, would be selected against. But if there were a situation of a different environmental condition, the susceptible mice would probably do better than the resilient mice that just seem to be maybe a little oblivious, a little less interested, concerned about what's going on around them. And so that's what I meant before, that the definition of what's adaptive and maladaptive really changes depending on the circumstance. But very few people have done that experiment because it's so difficult to do. The governments, NIH would not pay for that experiment, for example. I don't know about the MRC, but I know the NIH will not pay for it.
Amazing talk. Thank you so much. My question just relates to, I guess, the idea of, depression being caused by induced stress in these animal models and the application of that to human beings. If human beings, do develop depression as a result of prolonged stress from their environment, I guess what questions does that raise about societal or environmental changes being better avenues for, mass treatment for depression rather than medicinal interventions? Thank you.
Absolutely. So we have created for ourselves in our, quote, advanced cultures, high levels of stress that were not really selected for through evolution, right? So we think that the reason why we see these stress changes in mice and rats is because we have recapitulated situations that really groups of animals are not subject to this sustained chronic stress, right? In the animal kingdom, if you've ever watched the Discovery Channel or the Nature Channel, animals will either succumb to a predator or not, right? It's something that's decided pretty quickly. We, uniquely as humans, have done this to ourselves with, high levels of stress. Social media have dramatically worsened the ambient levels of stress and pressure, particularly on younger people. And so you are absolutely right that rather than medicating everybody, we should definitely work to alleviate those high levels of stress. I would be the first to sign on to that. And that's why I emphasized the importance of non-medication approaches. I don't know what it's like in Australia, but I'll tell you one thing. In the United States, it's hard to get these data because colleges don't like to talk about them, but somewhere around 30% of all of our college students are treated with a psychiatric medication at some point in their 4 years of college. Is it similar in Australia?
Yeah.
Yeah. This is really not good, right? Yeah.
Okay, one last question here.
Sorry, can I just circle back to the behavioural modification? Have you seen a difference in treatment responses by gender in humans rather than in mice, or in mice as well?
What do you mean by difference in treatments?
One like a teacher of boys, so I imagine running them is much better than getting up to
Oh, I see. Yeah. Right. No, absolutely. That's a great idea. No, we have, in general, you do not see a dramatic difference. There are published papers showing statistically significant differences in antidepressant responses in men versus women, but they're small differences. So by and large, as a psychiatrist, the antidepressant responses are similar in men and women. We see the similar thing in mice. But again, these antidepressants were all selected in an era when no one paid attention to sex differences. So I think we found them, because we found these medications because when you treat people, both groups respond, so you get a significant effect. I think what our evolving data are supporting is the idea that we will obtain more effective treatments, partly by focusing on sex-specific therapies, which is really quite novel for the field. I mean, that's something that has not been entertained, and I think it's really essential.
Well, thanks very much, Eric.
Thanks.
David Merson.
Thanks for having me.
Well, that was a stunning presentation. I'm reminded of my first contact with QBI. Its founding director, Perry Bartlett, had a conversation with me before this building existed, before the QBI existed. It wasn't so much a conversation, he was giving me a lecture, really. But basically, what he said to me is, he says, look, one of my fundamentals for this institution is going to concentrate on the science. And he was comparing it with other institutions who, I guess, looking at sort of just patient care type therapeutics. And Perry wasn't saying that science is more important than curing disease. What he was saying, or he enumerated to me, is that the best way of curing disease, if we understand the chemistry and the science of what's causing the problem, and then you can get some. And the illuminating thing about Professor Nestler's doing is applying that same principle to something which is extraordinarily complicated and difficult. And I know, I'm not sure if Perry's here, he'll be saying, I love it. So, yes, look, we are going to have some, everyone is invited to stay for some drinks and further conversations. But thank you so much for coming out. I'm sure you've stimulated a lot of thought and understanding, and certainly one thing is how damn complicated it is to do this sort of work. But it's wonderful that you're doing it, and I hope it's going to stimulate a lot more people to do it. Thank you so much.
Thank you.
And thank you.